They have also got intermediate filaments, and these intermediate filaments are desmin. And in case of the smooth muscles in the blood vessels, the the intermediate filaments are vimentin. Now the thin filaments and the intermediate filaments, they are attached to the dense body. This dense body is functionally similar to the Z disc, which is present in the skeletal muscle and cardiac muscle. We will discuss it later on. Now, on the basis of nerve supply, the smooth muscle can be divided into multi-unit smooth muscles and unitary smooth muscle. If the smooth muscles have good nerve supply, it is called multi-unit smooth muscle. And if they have not does not have a good nerve supply, they are called unitary smooth muscle. Now, skeletal muscle. So before going to the skeletal muscle, I would revise the smooth muscle again. The important points in the smooth muscle. The smooth muscle, they are fusiform in shape. The center lies in the nucleus, and they have got the gap junctions. So these three are important things. Now, the smooth muscle cell, in the cytoplasm, there are thick and thin filaments and intermediate filaments. The thin filaments and intermediate filaments, they are attached to the dense body, which is functionally similar to that of the Z disc present in the uh, skeletal muscle. Now, coming to the skeletal muscle. The skeletal muscle cell, they have got three layers of connective tissue: endomysium, perimysium, and epimysium. The skeletal muscle, they have got three layers, important layers. They are endomysium, perimysium, and epimysium. The endomysium is
عدنان صاحب ستاسو د اواز منځ منځ کې کټ کیږي غیب شي بیا راشي بیا غیب شي پکې تاسو
ها
لږ وګورئ چې دا څه مسله ده
خاجي
सर रिपीट कोई लगता है
ٹھیک صحیح ده مال بس یو منټ راکړي جی ضیاءالدین سے ہو हेलो
जी बस टिक दे आवाज सही दे राजी जी बस
आवाज ठीक है
कंटिन्यू करें
हाँ
आओ जो
हाँ so the skeletal muscle is surrounded by three layers of connective tissue. One is endomysium, perimysium, and epimysium. So each muscle fiber, it is surrounded by the endomysium, and that is loose connective tissue. The perimedium, the bundle of muscle fibers or the muscle fascicles, or the muscle bundles, they are surrounded by the perimedium, and then the whole muscle, it is surrounded by the epimedium. If you see here, this is the individual muscle fiber, and this individual muscle fiber, it is surrounded by the endomysium. Now the muscle fascicles, they are surrounded by the perimedium, if you see here, and the whole muscle cell, it is surrounded by the epimedium, if you see here. So the muscle, the skeletal muscles have three distinct layers: endomysium, perimysium, and epimysium. Now, how does the skeletal muscle look like? On light microscopy, as we already mentioned, that the skeletal muscles and the cardiac muscles, they have the striations. So here in the skeletal muscles, you can see on light microscopy, there are striations present. The light bands and dark bands, the dark pinkish bands and the light bands. So these are the striations. Now, how does what is the shape of the skeletal muscle? So the shape of the skeletal muscle, it is cylindrical shape. It has got cylindrical shape, and it is non-branching. If you see, if you discuss in the if you see to the cardiac muscle, they are branched, while the skeletal muscle and the smooth muscles, they are non-branching. So the skeletal muscle, it is cylindrical in shape, non-branching, surrounded by the external lamina, as we already mentioned, and the skeletal muscle, this is the skeletal muscle is multinucleated. So if you compare it to the smooth muscle and the cardiac muscle, in smooth muscle it has got one nuclei, while in the skeletal muscles they are multiple nucleated, which lie in the periphery. If you focus on this diagram, the nuclei lie in the periphery just beneath the cell membrane. And in case of the cardiac muscle, the nuclei can be either one or two in number, which lie in the center. So this is the comparison between the skeletal, cardiac, and smooth muscle in terms of nuclei. I would repeat it again. In case of skeletal muscle, the nuclei, they are multinucleated and they lie peripherally. In case of smooth muscle, it has got one nuclei and they lie in the center. In case of the cardiac muscle, the nucleus lies in the center, and the nucleus can either be one or two in number. So I would just show you the slide. If you see here in this slide, the nuclei, they are in the periphery. So this is in case of skeletal muscle. In case of smooth muscle, the nucleus lies in the center. In case of the cardiac muscle, the nucleus lie in the center, but nucleus can be either one or two in number. Now, I would repeat this slide again, that the skeletal muscle on light microscopy, it shows striations. It is cylindrical in shape, non-branching, and it is surrounded by the external lamina, and it is multinucleated, and the nuclei lie peripherally just beneath the cell membrane or the plasma lemma, sarcolemma. So I already discussed this slide. If you see here in this slide, the nucleus lies peripherally, and the skeletal muscle cell show the light bands and dark bands. So these are the light bands and these are the dark bands. Light bands and dark bands. Again, this is the diagram of the skeletal muscle, which shows this nuclei, they lie peripherally, and light bands and dark bands. So now coming to the cytoplasm of the skeletal muscle. If you remember from the previous slide, in the cytoplasm, there were thick and thin filaments. In the cytoplasm of the smooth muscle cell, there were thick and thin filaments. So mainly actin and myosin, and they also contain the intermediate filaments. And then the thin filaments and the intermediate filaments, they were attached to the dense bodies. And we discussed that these dense bodies were functionally similar to the Z lines of the
सर आवाज गायब शो
गरमंडे
सर जी आवाज निश्ता
plasm. Here have myofibrils, and the myofibrils, they appear like dark bands and light bands. They appear like dark bands and light bands. So if we focus here, these, they contain the actin and myosin. And the actin and myosin, they interdigitate between each other. So here we see these are the Z lines. To this Z line, actin is attached. Here if we see, this is the Z line, and to the Z line, actin is attached. And then we see M line. To the M line, troponin is myosin is attached. Again, I'm repeating. To the Z line, the actin is attached, while to the M line, myosin is attached. So the myosin is thick filament, and the actin is the thin filament. So here we have learned that the cytoplasm contains the myofibrils, and the myofibrils are cylindrical in shape, and they run parallel to each other. Now these myofibril, on microscopy, there are different bands, dark bands and light bands. So here I discussed that when there is a light band, it means it is actin, and if there is a dark band, there it means it is myosin. And actin is attached to the uh, Z line or Z disc, while myosin is attached to the M line. Now you focus here on the myosin. The myofibrils can be either thick and thin filaments. So the thick filaments are myosin, which consist of heavy chains and light chains. So I would just make it easy that each myosin molecule, each myosin is composed of two heavy chains and two light chains. So if you see here, this is there are two heavy chains and two light chains. The heavy chains has a thin tail and a big head. So this is a globular head, and this is the tail. So the myosin 2, it is composed of two heavy chains and two light chains. So if you see, if you focus on this diagram, these are the myosins. This is the myosin. Now, as I already mentioned, when there is a myosin, the myosin, the myosin on the microscope, it will show the A band. It will show the A band. And the actin, this area, it will show the I band. So the actin is interdigitating in the you see these are the actin filaments. They interdigitate in the myosin. So these are the areas which are deficient in the actin. So this area is called the H band, and in the H band there lies the M line. So I would summarize this slide again. You see these thick lines, they are myosin, and these thin lines, they are actin. Now, as I already mentioned, M line, you see this is the M line, and the actin, which is the thin filament, they are attached to the Z line or Z disc. So this is the one concept. The other concept is that the actin interdigitates in the myosin. So now on light microscopy, how will the light and dark bands be shown? So this thick filament, it will be shown as the dark band, and this dark band is called A band. So these will show these are the A bands, and now this area, this is the I band. I band means they show the actin. Now the area which is composed both of actin and myosin, this is the area. The area deficient of actin, that is called the H band. You see here it is written H zone. So this is only composed of the myosin, and between the H band there is the M line. So we have the A bands and the I band, and between the A band, in the A band there is the H band, and in the H band there is the M line, while in the I bands there are the Z lines. So these are the Z lines. Now coming to the thin myofilaments, the thin myofilaments is composed mainly of actin, tropomyosin, and troponin. So here if you see to the diagram, they have got two actin polymers, two actin, the molecules which are circular in shape. These are two strands of actin polymers, and they also have got the thread of or the fiber of the tropomyosin. This green line, this is the tropomyosin. So if you want to make it easy, in the actin, in the thin myofilament, they have double stranded actin polymers and single thread of the tropomyosin, and to the tropomyosin troponin is attached. To the tropomyosin troponin is attached. So you see here, this is the troponin, this green thread, this is tropomyosin, and this double helical circular structures, they are the actin polymers. And this is actually that actin which is shown as the I band. Now to make it easy, the myofibrils are surrounded by the sarcoplasmic reticulum, and the sarcoplasmic reticulum has got the longitudinal tubules and cisternae. So these are the longitudinal tubules, and they have they have the cisternae. So I think to make it easy, the sarcoplasmic reticulum, they have got the tubules and the cisternae. So that is sufficient. Now I would summarize the skeletal muscle, again, the skeletal muscle is surrounded by the three layers: endomysium, perimysium, and epimysium. The skeletal muscle have striations on light microscopy. They are cylindrical in shape and they are non-branching, surrounded by the external lamina and the skeletal muscle. They are multinucleated and the nuclei lie in the periphery just beneath the cell membrane. Then, if you, I discussed it already that the myofibrils, they have the light and dark bands. So if you see here, they are the dark bands and they are the light bands. Now in the dark bands there is the H zone or H band, and between the H band there is the M line, which has not been shown here, but I discussed it in the other slides. And in the light band there is the Z line. If you see here this line, this is the Z line. This is the diagram of the skeletal muscles showing the nucleus in the periphery and the light bands and the dark bands. Then here we discussed that the cytoplasm of the skeletal muscles have the thin and thick filaments, and the thick filaments are myosin, and thin filaments are actin. The thick filaments, the myosin, shows A band on microscopy, while the actin shows I band. And when we see the A band, in A band there is the H band and M line. This is the actin, sorry, this is the myosin. The myosin 2 is composed of actin and myosin. So this is the diagram. So you see this is the myosin. So here it will be shown as the A band, and this is the actin. It will be shown as the I band. And then I discussed that in the A band there is the H band and M line. So this line, this space, this is the H band, and between the H band there is the M line. ठीक से
دا ریپیټ کې ورته ستله د کې
clear
आवाज
है
نه و کنه
ठीक है दे रखा था okay so each muscle fiber, the skeletal muscle fibers, they are surrounded by the sarcoplasmic reticulum, and the sarcoplasmic reticulum, they have two main parts. One is longitudinal tubules and the other are the large cisternae. And these cisternae, here there are cisternae, they are attached to the T tubules. The T tubules are actually the invaginations of the cytoplasm of the skeletal muscle. So I would repeat this slide again. The sarcoplasmic reticulum consists of tubules and cisternae. So the tubules, here are tubules and these there are the cisternae, and this cisternae is attached to the T tubule, which is actually the part of the cell membrane of the skeletal muscle. Now, coming to the cardiac muscle. So, as we have already mentioned that the cardiac muscle is striated. So, in terms of the striation, it is just like uh, the skeletal muscle. Here, if you see, these are the striations, the dark and light bands. The cardiac muscle is the involuntary muscle, and it is supplied by the autonomic nervous system. Now, the other thing regarding the cardiac muscle is that they lie in chains. The cardiac muscle lies in chain. If you see here, these are the chains. This is one cardiac muscle cell. This is the other cell, but they lie in chain, and they are attached to each other by discs. And these discs are called junctional complexes. They are attached to each other by the junctional complexes. So I would repeat it again. The cardiac muscle, it is a striated muscle, and the striation means that it has got the dark and light bands. It is an involuntary muscle and supplied by the autonomic nervous system. Now, in case of the skeletal muscle and in case of the smooth muscles, they did not lie in the chains, but here the cardiac muscle lie in chains. You see here, this is the one cell and this is the other cell, and they are connected to each other by the junctional complexes. Now, these chains, they are surrounded by the network of collagen and reticular cells. And this network of collagen and reticular cells, they resemble the endomysium. They resemble the endomysium of the skeletal muscle. So you just need to remember that each muscle cell, they are surrounded by the network of collagen and the reticular cells. Again, if you see here, this is the cardiac muscle, and they have got the chains, and these chains are formed by the different cells, and they are branched. If you see here, this is the skeletal muscle, which is not branched. These are the smooth muscle cells, they are not branched. But here, this cardiac muscle, they are branched, they lie in chains, and they are connected to each other by the junctional complexes. Take. Now, we discussed already this slide. So coming to the other slide, here if you see, I already told you that the cardiac muscle chains, they have they branch one thing, and they have intercalated discs or junctional complexes. So you see, these are the intercalated discs or junctional complexes, and the nucleus lie in the center. Now, regarding again the nucleus in the cardiac muscle, they lie in the center, and they can be either one in number mostly, but it can be two in number also. Now, the sarcoplasm or the cytoplasm of the cardiac muscle, it consists of myofibrils, and these myofibrils, they look like the same as in case of the skeletal muscles. So how does the myofibril shows? They will show the A band and the I band. So A band, in A band there is the H band, and in the H band there is the M line, while in the I band there are the Z lines. So this is same as that in the skeletal muscle. The cytoplasm of the cardiac muscle, it has mitochondria, which is abundant in number, and it has also called atrial granules or atrial natriuretic factor. And this ANF, the function of ANF is that it causes diuresis in the in it causes diuresis. So this is the another function, and this ANF is present in the, especially in the cardiac muscles of the atrium, in the atrium. So I would repeat this slide again, that the cardiac muscle, it has a nucleus which has a nucleus which can be one in number or It can be 2 in number also. And the cytoplasm of the cardiac muscle, it has myofibrils, and these myofibrils are structurally the same as that of the skeletal muscle. It has abundant mitochondria, and the atrium of the cardiac muscle, they have also got atrial ganglia which have A and F. Now, talking about the intercalated discs again, these are the transverse bands. You see, these are the transverse bands, and they are composed of the junctional complexes. They are composed of the junctional complexes. Now, this junctional complex is composed of the fascia adherens, desmosome, and the gap junction. What is this junctional complex? These are different kind of These are the different kind of cell junctions, and these cell junctions are fascia adherens, desmosome, and the gap junctions. Because of the fascia adherens and desmosome, the skeletal muscles, the cardiac muscle cells, sorry, the cardiac muscle cells, they are tightly adherent to each other, while the while the function of gap junction is that they communicate the cardiac muscle cells, the cardiac cells, they communicate with each other, and because of this communications, the cardiac muscles, they contract in the syncytial fashion, in a syncytial fashion. Again, the tubule. The tubules are the invaginations of the cell membranes of the cell membranes of the cardiac muscle, and the difference between the T tubules of the skeletal muscle and here in the cardiac muscle is that the T tubules here in cardiac muscles, they are large in diameters, and they lie at Z discs. You know where is the Z disc? The Z disc or Z line lies in the I band. So, but the T tubules present in the skeletal muscles, they lie at the junction of the AI junction. So you just need to remember that the T tubules here in cardiac muscles, they have large diameter, while the T tubules in the skeletal muscles, they have the small diameter. Now coming to the sarcoplasmic reticulum or the endoplasmic reticulum of the cardiac muscle. They have got the longitudinal views as present in the skeletal muscle, but they don't have the large terminal cisternae cristae, sorry, as present. They have got the small terminal cristae. So this was all about the skeletal, cardiac, and smooth muscle cell. If you have any questions, just write down.